So this is a presentation actually, or it's adapted from something that was made by a friend of mine. He was a chemist at Pfizer, working on drugs for central nervous system. And he designed like antipsychotic drugs and things like that. And this is a little presentation that he made that just, I think, makes it kind of clearer about what does it actually mean if you have a ligand and a receptor binding, and how the heck does that actually cause an activation of a G protein and start the second messenger pathway? And what is going on with exogenous ligands? How is it that caffeine or magic mushrooms or something like that will affect your brain? So let's take a look. So this particular presentation is about serotonin. And this one's going to get in particularly looking at one of the serotonin receptors that's found responsible for most of the psychedelic drugs. So serotonin, a classic neurotransmitter used by many neuron cells throughout the brain. This is just showing lots of neurotransmitters. And I should also mention while we're here, another word for serotonin is 5-HT, 5-hydroxytryptamine. So these will be used interchangeably. It'll either be using the word serotonin or using 5-HT. Those are the same thing. That's like saying, a dog and a pooch, or a cat and a feline or something, right? Serotonin and 5-HT is just two ways to say the same thing. Okay. So he's saying here there are 14 known serotonin receptor subtypes. You've already heard briefly, I talked about a couple of the, acetylcholine subtypes. We talked about muscarinic versus cholinergic. When we get to the, norepinephrine, adrenaline, noradrenaline, we're going to have a whole bunch of different adrenergic types of receptors, the alpha 1, alpha 2, beta 1, beta 2. It's the same thing with serotonin. There's a whole bunch of different subtypes of serotonin receptors. This particular presentation is going to focus on this one, the 5-HT2A receptor. This is one that gets quite a bit of interest because if you look across all the different psychedelic drugs and try to figure out what do they have in common, binding this receptor is one of the things they all seem to have in common. Right? If you look at something like LSD is what they call a very promiscuous ligand. It binds lots of things. It binds dopamine, it binds serotonin, but the thing that is common across all the psychedelics is they all seem to share this one. So that makes this one particularly interesting. So that's what this presentation is going to look at, is this. And again, what is a receptor? A receptor is made out of what kind of biomolecule?
Protein.
It's a protein. It's a membrane protein. So we're going to have to get into the detail of the protein that makes up the membrane. We're also going to have to get into the detailed chemical structure of the ligand, the serotonin molecule itself. And then we're going to see how they come together in binding. And so we'll see what binding really means on a more detailed level. But in this process, we're going to be reviewing chemical bonds, and we're going to be reviewing concepts that we've been talking about. So I think this is going to be interesting and useful. But like I was saying, you do not need to know that there are 14 known serotonin receptor subtypes for your exam. Don't get involved in any of the details. Just think about how it reviews the things you already know. Okay, it's not unusual. Most receptors have at least 2 or more subtypes. So now he's like, in order to go forward, we have to get small. I don't know if you remember, what's his name? Steve Martin. Let's get small. So we're going to go down the atomic level, and again, it's going to be looking at the stuff you already know. We're going to be looking at non-polar stuff, things that do not have charge and tend to like cluster together. Hydrogen bonding, where you have local charge imbalances, which then attract through the electrostatic attraction. Ionic bonding, where something actually donates an electron to something else, and then you have the opposite charges that pull in. So we're going to see all of these being important. And let's continue here. Again, nonpolar things, basically no charge imbalance. Hydrogen bonding, where you have local charge imbalances and therefore electrostatic attractions. Ionic bonds, where you actually totally give up one electron to something, the other thing accepts it, you get 2 ions, which then pull. This is showing ionic bonds don't just have to be between 2 atoms like sodium and chlorine. This is about the amino group, and so you can have different things that are connecting up. So here, like the carboxyl and the amine are swapping electrons to create a ionic bond. These are parts of larger molecules. So when we see our protein, that is the receptor, some of those amino acids can contribute to ionic bonds. When we look at the serotonin molecule itself, it can also contribute to ionic bonds. So if we look at serotonin, let's take a look here. What do we know about, so let me get a different color because I know. What do we know about serotonin, if you have hydroxyls hanging off of some molecule? What kind of bonds do those usually end up participating in?
Hydrogen bond.
Hydrogen bonding, right? Remember, that's when we talked about how sugar dissolves in water, how sugar does hydrogen bonding. When you drew that picture in your first lab, I talked about how those hydroxyls hanging off are local charge imbalance. Right, so when you see a hydroxyl hanging off, it should go bing, that's hydrogen bonding there. This nitrogen in this little ring here, that also is going to participate in hydrogen bonding. It causes a local charge imbalance. So we have parts of this molecule that like to make hydrogen bonds. Again, this is serotonin, right? Any vertex that is not actually defined as a carbon, right? These are carbons at each of these corners. All right, so we have parts of it like to make hydrogen bonds. This part here, this part of the ring where we just have kind of more this kind of symmetry, this tends to be more of a local hydrophobic area on the molecule. So we have another section, we have sections that are more polar that make hydrogen bonds, another section that tends to be more non-polar. And then we have this amino group, and this amino group actually likes to donate electrons to make ionic bonds. So we have one part that wants to make ionic bonds, a couple of parts that like to make hydrogen bonds, and then another part that tends to not like to interact with charge imbalances at all. And it's also important that this has a very specific shape. So it's this part that likes to make the hydrogen bonds, this part that makes the ionic bonds. So when we talk about the actual binding, like where it binds to the receptor, we're going to have to find something that it can fit into that has a pocket, and where this part of the pocket likes to make ionic bonds, and this part of the pocket likes to make hydrogen bonds, and this part of the pocket is kind of nonpolar, and this part of the pocket, it also likes to make hydrogen bonds. So when we talk about binding, we're going to find the receptor has kind of the complement to both the shape and the chemical properties. And that's what binding is going to be. This thing's going to settle into its perfect mate, so to speak. Does that make sense? All right, so let's continue on this. Hydroxyl forms hydrogen, aromatic in the ring, amino group can make an ionic bond, and the nitrogen forms a hydrogen bond, right? And when we talk about exogenous ligands, if we have another molecule that's not serotonin and yet has these basic properties, it's got a good chance of binding the same receptor, right? Because it's going to be a mimic. It's going to like have the same basic look and feel. That's what, if you give somebody morphine, it's not a naturally occurring opioid, but it looks close enough that it gets in the receptor and binds it. You give somebody, psilocybin, magic mushrooms, it's not exactly serotonin, but it's close enough that it can get into the receptor and bind there, which we're going to see in more detail in a few moments. We'll look specifically at magic mushrooms as we continue here. All right, so that is the ligand. The other thing we need to look at is the protein, that's the receptor. And again, this is just reviewing the stuff you should know. So this is, I feel we're actually reviewing things that is going to be useful for you to review for your exam. Proteins are made out of amino acids. The amine group, the carboxyl group, and that side chain that we talked about, the side chain can make hydrogen bonds or make ionic bonds or be nonpolar, right? We talked about all these different kinds of amino acids, and each kind of amino acid has different kinds of chemical properties. That's what makes it important to hook up one amino acid versus another. What do we call, here's 20 naturally occurring amino acids. And again, they're grouped here in terms of their different properties, which we'll talk about a little more later. What do we call this part of the structure if we just hook them up with peptide bonds and make a big long chain of amino acids?
Polypeptide?
Say it again?
Polypeptide?
So polypeptide just means less than 100. But what do we call this part of the structure? We talked about different versions of, or different levels of structure for a,
primary?
This would be primary. The primary structure of a protein is just which amino acid is hooked up in which order. Right? And again, these R groups, these side chains can be different depending on which amino acid it is. They might make hydrogen bonds or might make ionic bonds. What's the next level of structure for a protein?
Secondary?
The secondary. And what does secondary structure of a protein look like?
Has hydrogens.
What's that?
Doesn't it have hydrogens in between the bonds or something?
I'm not quite sure what you mean, hydrogens between the bonds.
In the chain, isn't there's another element added in? No? Never mind.
Either alpha helices or beta sheets.
Yeah, the alpha helices and the beta pleated sheets, and they're held together by hydrogen bonds, the electrostatic attractions. There's no extra pieces. It's just hydrogen bonding between different parts of the big molecule. So we'll get there.
So secondary is what?
The alpha helices and the beta pleated sheets.
But those are formed due to the hydrogen bonds.
They're formed due to hydrogen bonding between
That's where
different
I was
parts.
getting confused with something with hydrogen. Okay.
Yeah, and then what's the next part, remember, was the tertiary structure, where you have the more big overall folding with the hydrophobic, hydrophilic interactions and all that. What was the tertiary structure?
3D folding.
Say it again.
3D folding, when it starts to like fold and create a shape.
No, that was that was tertiary. And then what about if we go, yes, yeah, you are correct. So it's where you have that more big 3D folding and where you have usually often a functional protein at that level. What about the quaternary structure, the fourth degree?
Multiple of the tertiary?
Yeah, you need one or more, two or more of the subunits that work together to have a functional protein. All right, so now this part is interesting. So he says, okay, we know that a protein is made out of a bunch of different amino acids hooked together, in some order determined by the DNA code. So he says, is a protein like, a little chain of colored beads? And he's like, not really, because these beads are all substantially similar. They just have different colors, but amino acids are actually pretty different from each other. We talked about those side chains can do very different kinds of things, make different kinds of bonds. So he's like, or maybe more like a charm bracelet. So these look a lot more radically different from each other than these beads. But he says, like, this is still not a very good way to understand a protein, because it's not just what they look like, it's how they interact, what kind of bonds they like to make, what different amino acids like to do, if they want to be polar or non-polar. So he says, it's really better to think of it like a tool belt. Because you think about a tool belt, these different sections of the tool belt are very specialized to have very different kinds of interactions. If you take the hammer and try to put the hammer in the thing that's made for the tape measure, it's not going to fit. It's not going to sit there. It's not going to bind. If you take the tape measure and try to put it on this thing made for the hammer, it's not going to stick. It's just going to fall off. So this idea that in a protein, the different amino acids have very specific kind of things they like to connect to, or different kinds of chemical bonds they like to make. So that's going to be important when we look at the receptor. The receptor is going to be made out of amino acids that like to make different kinds of bonds. And as I was mentioning earlier, hopefully they're going to be set up in just the right way to fit and mate with the ligand, with the neurotransmitter that is going to bind with the receptor. So does that make sense? This idea of a tool belt, each amino acid makes very specific kinds of chemical bonds. All right, continuing on. So it's just saying basically some of these amino acids have non-polar side chains. Some of them make polar hydrogen bonds. Some make polar negative ionic bonds. Some make positive polar ionic bonds. So different amino acids like to make different kinds of bonds, which is just, reiterating what I've just keep yapping and saying. So now we're ready to start assembling the actual receptor. Again, it's going to be this thing with, these are each of the colors here are representing an amino acid. I can, in this picture, the membrane would be here. There are going to be 7 transmembrane helices. These are just alpha helices that are going across the membrane. These transmembrane helices are going to be mainly what kind of chemical property? If they're going to, if that part's going to actually stay in the membrane.
Hydrophobic.
Hydrophobic, exactly. That's why it's going to stay there. And then we have these parts that are going to be outside the cell that are going to actually bind with our receptor. And then we're going to have the parts inside the cell that are going to be interacting with our G protein. These are called the cytoloops, the parts of the protein that are inside, the exoloops, the part on the outside that are going to interact with the receptor, and then these transmembrane helices that are actually embedded in the membrane. So that's a classic receptor. And this is what the serotonin 5-HT2A receptor looks like.
Question.
Uh-huh.
Which side is the cytoplasm? The side with the G protein?
Yes.
Okay, thanks.
Okay, here he's just showing the alpha helix in more detail. Basically, every third amino acid does a hydrogen bond with the amino acid below it. So that's creating these transmembrane helices that I kind of just mentioned. These exoloops are the parts of the protein that are going to be outside the cell. Cytoloops are the parts that are inside the cell. You know, this is an inform. You know, we have these transmembrane helices. This is where my serotonin is going to come down and dock. Down here is where the G protein is going to be and get activated. And basically, as the serotonin binds at the top, it's going to tweak and pull on this thing in such a way to change the shape of the bottom to activate the G protein. It's kind of what I mentioned last class, but now we're just seeing it in more detail. Okay. Right, so here, this is just kind of the picture that you've seen. Again, I will mention one more time, just because probably you can't say it too many times. We're going to have our ligand binding up here. As that happens, it's going to tweak the shape of this just right to activate and catalyze the G protein. And once the G protein is affected or turned on, it's going to go and, you know, in the case of the example I showed last time, turn on adenylyl cyclase, or in other pathways, it might turn on some other enzyme like phospholipase C and end up releasing calcium in the cell or something. So this is how most of the CNS receptors seem to be hooked up. The receptor binds, tweaks this thing, which activates the G protein, and then the G protein goes off and initiates the second messenger pathway. What we're going to do right now is just look at detail. And again, way detail you don't need to know, but I think it's kind of fun for you to see what does it actually mean if a receptor binds a ligand. So I'm going to show you that right now. Again, don't worry about the details. The details are not the point. The point is kind of getting a feel for things. All right, so here's this membrane protein, which is a receptor. This is what it looks like as, so I am Mr. Serotonin drifting down towards the cell, and I see this big, crazy kind of structure full of different amino acids. And some of them like to make hydrogen bonds, some like to make ionic bonds, and they're also held together in this big form, in this big, crazy three-dimensional folding. So what this is here now is we are looking at the serotonin itself, kind of drifting in. This green thing is the model of the receptor. And this is all this kind of molecular modeling, looking at what actually happens when the serotonin drops down into the receptor site. And again, remembering there's parts that like to make hydrogen bonds, part that is more nonpolar, part that likes to make ionic bond. So this is going to drop into the pocket. Again, on the third transmembrane helix, the amine group of serotonin forms a strong ionic bond with an aspartic acid and also a hydrogen bond with an adjacent serine. So we have our amine, so the amine group of the serotonin, which is right here, is hydrogen bonding with these two places, these two amino acids. On a transmembrane helix 5, a serine forms a hydrogen bond with the nitrogen group. So here's the nitrogen over here, and it's making an ionic, a hydrogen bond with another one of the amino acids that's part of the protein. On transmembrane helix 6, 2 phenylalanines and the tryptophan make a local hydrophobic pocket where that hydrophobic part sits. And finally, the hydroxyl that was hanging off of the ring makes a hydrogen bond. Where's the hydroxyl? There's a little hydroxyl. This little hydroxyl here is making a hydrogen bond with the serine up here. So we got a little hydrogen bond action going. So basically, this little thing is sitting in the pocket where this group is making hydrogen bonds here. This group is making hydrogen bonds here. This is in its local hydrophobic pocket. This is making hydrogen bonds down here. And in all of these cases, it fits in, and as it fits into its little pocket, it kind of yanks and pulls a little bit. That's going to be an important part of this story as well. As it makes these little attractions, it's going to yank on these helices and change their conformation slightly. Right? Imagine you kind of are sitting into your pocket, and as you really settle in, you just pull everything tight against you. So that's what's happening here. This serotonin settles in. As it really settles into its pocket, it pulls everything tight. And as it pulls everything tight, the membrane transmembrane helices shift slightly. As they shift, that is what changes the shape on the cytoplasm side and turns on the G protein. So does that make sense? And again, once the G protein is activated, a typical thing is turn on adenylyl cyclase. And once you turn on adenylyl cyclase, what happens next?
Second messenger.
Yeah, which one in particular?
Cyclic AMP.
It makes cyclic AMP. Once you have cyclic AMP, what does cyclic AMP often do?
Does it bind with an enzyme or something?
Yeah, what enzyme does it turn on?
Kyanase?
Exokinase.
The
Kinase A.
protein kinase. Protein kinase A in particular. Remember the hexokinase, and that phosphorylates sugars. That's something totally different. That was in glycolysis. And then what do the protein kinases do?
They'll
Phosphorylate
phosphorylate some enzyme or do whatever their ultimate purpose in the cell, in that specific cell is.
Yeah, so those phosphorylate things, and in doing so are typically either opening or closing channels as they phosphorylate a protein, or they might be turning on or off an enzyme, phosphorylating another kind of protein, right? So, make sure you understand, like I mentioned last class, second messenger pathways are not going away. They're going to be at the core of all sorts of stuff. So make sure that they ultimately make sense. What ultimately helps turn off the second messenger pathways once it's been triggered? In this,
Phosphodiesterase.
yeah, exactly. That PDE, the phosphodiesterase, which breaks down the cyclic AMP, so makes the pathway eventually stop. Talked about that's how Viagra works. Viagra works by blocking the phosphodiesterase. So the pathway that turns on the dude's erection keeps going and the dude stays hard. So, you know, you definitely want to kind of think about these second messenger pathways for the class in general and definitely for your exam. All right. So the last thing I'll use just to tie this up here is this idea of an exogenous ligand, right? Lots of different drugs, whether they are pharmaceutical things like, like morphine or like albuterol or like, Xanax, or whether they're recreational drugs. Here, psilocin. Psilocin is from magic mushrooms. It's when you eat like a hallucinogenic mushroom, there's psilocybin, which as soon as it gets in your digestive tract, gets converted to psilocin. So this psilocin is the active ingredient ultimately in your body that is going to cause the experience of eating a psychedelic mushroom. So let us take a look. They look awfully similar, right? Oh my God. The hydroxyl is shifted a little bit. It's off one spot on the ring. We have these methyl groups hanging off of the nitrogen instead of just hydrogen. So it's a little bigger and more complicated here. But it's not shocking, hopefully. If you look at these two things, it's hopefully not shocking that psilocin has a pretty good chance of causing mischief in the serotonin receptor. Wow. Hold on, I just had this, I have a disk that keeps dying. Hold on, I need to turn this off, close this. All right, so does that, is that kind of intuitive as to why serotonin and psilocin have, when you have an exogenous ligand, it's going to have both a physical structure as well as kind of a chemical bonding kind of proclivity that are going to be similar to the endogenous ligand. And that's why it's going to fit into the receptor and be able to bind. Maybe if it binds and excites the receptor, what do we call that?
Agonist.
An agonist. If it gets in there, but it kind of just jams it up and doesn't actually turn it on?
Antagonist.
An antagonist. Psilocin actually ends up being a partial agonist. It turns it on, but not quite as strongly as the native one. Right, so if I continue on this, it basically shows how psilocin gets in there and it has many of the same interactions as the native serotonin, but it doesn't go in quite the same way, because those big methyl groups on the amine, it can't get as close. It doesn't make as strong hydrogen bonds. It doesn't pull as tightly on the helix, so it doesn't change the shape quite as dramatically as it would have otherwise, which is part of the reason it's going to be a partial agonist, because it's not going to get pulled into its fully functional shape. But that's basically the idea of, let me stop sharing there. So does that help make the idea of exogenous ligand be a little clearer? Right, in fact, maybe let me, before we leave this completely, let me just get, hold on. Images. Here, I'm going to show you another. I'm going to share screen again. So here's caffeine and here's adenosine. So caffeine caffeine, most people know caffeine. That's the stuff that you find in coffee or tea. Tends to make you not get so tired. It turns out, you look at this thing, it has this section that has the same basic shape. It turns out it can get into the adenosine receptor. Adenosine acts as a neurotransmitter. Right? Adenosine is just another molecule that we talked about. Adenosine as part of ATP or part of DNA, but anything can be anything, right? Like in fact, I often talk about like, what is, if I have a pencil, what is it? It's a thing I can write with it, or it's actually a thing that helps me clean the wax out of my ears, or helps me pick my nose, or it's a murder weapon. You can stab somebody to death with it, right? It's like, what is this? It's a thing. And depending on the context and how you use it, it's doing different jobs. So adenosine in your nervous system does the job of a neurotransmitter, just in case you're kind of wondering, like, what's it doing there? But if we kind of get back here, adenosine receptors actually make you tired in your central nervous system. So the caffeine is actually an antagonist to the adenosine receptors. The caffeine gets into the adenosine receptors, and as adenosine builds up, which would normally make you tired, it can't get into the receptor, and therefore you stay awake. Right? But it works. The only reason adenosine or caffeine works is because it has a shape that can fit in to the receptor and cause an action there. So, LSD. If you look at LSD and serotonin, it also has that, just like psilocin, fit because it had a shape. This, LSD is a bit more of a complicated thing, but it still has that section that can get into that serotonin receptor and bind and cause activity. Like I said, LSD in particular has lots of other things it binds to and lots of other receptors it can activate. But for the psychedelic or a main part of the psychedelic effect, it seems important that it has a shape to get into and activate a 5-HT2A receptor. So just kind of bringing home that point that an exogenous ligand does its magic by having a physical shape as well as chemical properties that mimic the naturally occurring endogenous ligand, and therefore it can get into the receptor and bind it the same way like the naturally occurring ligand could get in there and bind and therefore cause some effect. Does that make sense? So again, hopefully this is useful, both in kind of getting more insight into what receptor binding is, but also reviewing a lot of the stuff, like why we've been caring, why we have put all this energy into what is a hydrogen bond or an ionic bond, or what is a protein, what is an amino acid, what is, because you can't understand what it means to have serotonin binding a serotonin receptor unless you understand the basic chemistry.